All right, I hope you're ready for monkey human pizza delivery drivers because China just made a historic study where they gave birth to a chimeric primate, which is a cross between a human and a monkey. I have the Nature article here and the medical journal that published the study. So let's break this down. This hybrid baby monkey is made of cells from two embryos. The monkey's creation paves the way for scientists to use chimeric primates to study human diseases. Yeah, I wonder what else they're going to use it for. You can see right here it's a bit green, and that's from the green fluorescent protein, which we'll get into later. But that's a byproduct of this experiment. Scientists have created an infant chimeric monkey by injecting a monkey embryo with stem cells from a genetically distinct donor embryo. The resulting animal is the first live-born chimeric primate to have a high proportion of cells originating from donor stem cells. The finding, reported today in Cell, opens the door to using chimeric monkeys, which are more biologically similar to humans than are chimeric rats and mice, for studying human diseases and developing treatments, says stem cell biologist Miguel Esteban at the University of Chinese Academy of Sciences in Guangzhou, a co-author of the study. All right, we're going to head over to Cell. I'm just going to read a few sentences from here to sum it up. But in this study, we systematically tested the effect of various human PSC media, that's pluripotent stem cells. I have a little summary of it right here. Pluripotent stem cells are a particularly potent type of stem cell that normally only exists during the embryonic development. What makes pluripotent stem cells so potent is their ability to form all three of the basic body layers, the ectoderm, endoderm, and mesoderm, and even germ cells. Evidently pretty potent. So they took these human pluripotent stem cell medias on monkey embryonic stem cells and optimized the protocols for injection into early monkey embryos in the in vitro culture of the injected embryos. That means on a dish, basically. They found that 4CL, that's basically just an enzyme, produces an improved monkey naive pluripotent state for the survival of the monkey donor pluripotent stem cells inside homologous blastocysts. Furthermore, they established thorough tissue characterization involving green fluorescent protein cell counting. I have pictures of that. I'll show you that in a minute. Among other things, using these approaches, they demonstrated a high degree of chimerism of monkey embryonic stem cells, both in prolonged embryo cultures in vitro and during gestation in vivo, yielding a live offspring that displayed 20 to 90% of donor embryonic stem cells in chimeric tissues, including the gonads and placenta. Our result represents a proof of principle demonstration that chimera with high embryonic stem cells contributions can be generated in non-human primates through early embryonic complementation with homologous pluriponic stem cells, paving the way for future generation of genetically modified non-human primates with gene editing PSCs. All right. These are some of the pictures. These green ones, let me try zooming in. That is the green fluorescent proteins. And they're just comparing the different types of cells in here between the human and non-human, right? Pretty fascinating stuff. Okay, let's get back to this article. But the monkey chimera had to be euthanized when it was only 10 days old because of the hypothermia and breathing difficulties, highlighting the need for further optimization of the approach and raising ethical concerns, says researchers. Chasing chimeras. Scientists have long thought to make animal chimeras using embryonic stem cells, which are derived from an embryo's inner region and can develop into a wide variety of tissues. Such stem cells can be genetically edited before being added to a recipient embryo. For example, stem cells carrying genetic mutations that have been linked to a particular disease could be added to embryos without those mutations, This would allow scientists to study how stem cells carrying the mutations affect physiology and health. In earlier chimeric monkeys, just 0.1 to 4.5% of the cells in organs such as the brain, kidney, and lungs were derived from donor stem cells. Because the contributions were so small, these chimeras were unsuitable as models for human disease, says Esteban. To produce a chimera with a larger contribution, Esteban and his colleagues created recipient embryos by collecting eggs from female cynomolgus monkeys and fertilizing the eggs. Meanwhile, the researchers extracted embryonic stem cells from 1-week-old embryos and genetically edited the cells to display a green fluorescent signal. To grow the stem cells in the laboratory, the team fine-tuned the nutrients and growth-promoting proteins in the liquid in which the stem cells were grown. They then injected up to 20 stem cells into each of the recipient embryos, yielding 74 chimeric embryos with a strong fluorescent signal. Okay, they have a low pregnancy rate. I'm going to skip that part, but the platform for growing human organs. This work is both impressive and commendable, says stem cell biologist Irene Aksoy at the Stem Cell and Brain Research Institute in Lyon, France, who was not involved in the study. The method might be used to grow human organs in pig or non-human primate tissues, says developmental cell biologist Shukrat Mitolipov, director of Oregon Health and Science University in Portland. If we can delete the genes encoding for, say, the kidney in a large animal, such as a pig or primate, we can introduce human cells to produce that organ instead, he says. But he adds that using human animal chimeras for organ collection, especially if human embryonic stem cells contribute to the nervous system, brain, or reproductive cells, comes with many ethical concerns. Obviously, that's the biggest question here, right? How ethical is creating a human monkey chimera? I mean, do you want an Uber driver that's a human and monkey? Okay, all jokes aside, yeah, is it worth harvesting these things if it means you can get an organ from them, or testing all these different types of experimental drugs on them before they hit humans? Something just seems so wrong about it. And with the increase of computational exponential computing powers with machine learning and AI, like, these algorithms are running all the time to find the best way to do this. And they made a huge leap today by having over half of these cells being human, or at least the targeted cells. That's what it sounds like. So, pretty significant, actually, and it's only going to get more efficient from here. So, you already know that somewhere deep in China, somewhere deep in Russia, or, other countries perhaps, they're a little bit further ahead than what's being reported in Nature, I'm sure. I mean, just think about what's not being reported in this topic. And of course they would want to hold that close to them and not share it. So anyways, just thought I'd read through that. Hope it was interesting. That's your news for the day. And yeah, stay curious.